A study by the MRC Laboratory of Medical Sciences reveals that cells in different organs selectively express maternal or paternal X chromosomes. This variation, shown in both human data and mouse models, is driven by cellular competition, affecting organ development in biological females. Research reveals that selective expression of maternal or paternal X chromosomes varies by organ, driven by cellular competition. A new study published in Nature Genetics by the Lymphoid Development Group at the MRC Laboratory of Medical Sciences has revealed that the contribution of cells expressing maternal or paternal X chromosomes can be selectively skewed in different parts of the body. The study leverages human data from the 1000 Genomes Project combined with mouse models of human X chromosome-linked DNA sequence variation to advance our fundamental understanding of development in biologically female individuals who have two X chromosomes. Until now, it was thought that the usage of maternal and paternal X chromosomes was similar throughout the body. The new work shows that this is not always true, and that different organs may be skewed towards using either maternal or paternal X chromosomes. The work also reveals the process driving this skew: competition between cells expressing either one or the other X chromosome. In some individuals, cells in organs such as the heart mostly use the X chromosome from one parent, whereas immune cells almost exclusively utilize the X chromosome from the other parent. This provides an important step forward in understanding the underlying principles and mechanisms of development in Roman 20 individuals. Biological females inherit two X chromosomes, one from each parent, along with all the other genetic material that builds and sustains the body. But despite the presence of both parental X chromosomes, only one X chromosome is actively expressed in any given cell. Since the DNA sequence of each X chromosome has genetic variations, each cell effectively chooses to express a set of unique characteristics derived from either one or the other parent. Researchers realized that when cells chose one of their two X chromosomes over the other, they also chose which set of genetic variants to express. As a result, individual cells express distinct genetic variants. Researchers are now working to find out more about how X-linked genetic variants shape organismal development, and whether selective X chromosome usage in specific tissues may affect the likelihood of certain conditions later in life. The researchers focused on a specific gene on the X chromosome called STAG2. They found that cells with a genetic variant of STAG2 failed to develop into immune cells called lymphocytes in females that carried variant STAG2 on one X chromosome and the common reference version of STAG2 on the other X chromosome. By contrast, cells with the same variant of STAG2 were fully competent to form lymphocytes in XY males with a single copy of the X chromosome, or females in which both X chromosomes carried the variant. The researchers concluded that what prevents variant cells from forming lymphocytes is not the variant as such, but the presence of cells expressing the reference version of STAG2. This shows that cells compete for permission to form specific cell types within the body. The findings reveal a new aspect of X-linked diversity not previously appreciated, that interactions between cells can shape the contribution of X-linked diversity to specific cell types and tissues. Even if cells expressing reference STAG2 or compete to form the blood, cells expressing the variant may predominate in other parts of the body. These findings reveal a previously underappreciated aspect of X-linked diversity, where interactions between epigenetically diverse clones can shape the contribution of X-linked genetic diversity to specific cell types and tissues. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.